The overall goal of this procedure is to transform yeast cells with an arrayed library of plasmid DNAs. This is accomplished by first growing up an appropriate amount of yeast cells. The yeast cells are then treated with lithium acetate to make them competent for DNA transformation. Next, transform the competent yeast cells by mixing them with DNA. The final step of the procedure is to select and analyze transformants. Ultimately, results can be obtained that show the effects of the plasmid DNA on yeast phenotypes through yeast spotting assays and/or microscopy.
The main advantage of this technique over existing protocols, such as standard yeast transformations, is that it allows for the transformation of a large number of different plasmids into yeast cells in an automated fashion.
This high-throughput yeast transformation protocol is designed for 10 96-well plates, but can be scaled up or down accordingly.
For larger scale experiments, the key to success is to keep all plates and reagents organized once the experiment is underway to avoid confusion.
A biorobot rapid plate liquid handler will be used in this protocol. To begin, aliquot 5 to 10 microliters of plasmid DNA from an arrayed yeast plasmid library into each well of the round-bottom 96-well plates. Place the 96-well plates uncovered inside a clean bench fume hood overnight to dry. Next, inoculate 200 milliliters of yeast peptone dextrose, or YPD media, with a query strain in a 500 milliliter baffled Erlenmeyer flask. Incubate overnight at 30 degrees Celsius with shaking. On the following morning, dilute the query strain to an OD 600 of 0.1 in 2 liters of YPD media.